Well, thanks a lot, Hugh. It's a great pleasure to be here. Congratulations to the first two speakers. Really enjoyed these presentations and look forward to Phil's coming up to wrap up the day. And it's a delight to be here today. So what I thought I would do, we have a few different activities involved with the CCHF, but I wanted to tell you about a collaborative project between me and David Sherman here at Michigan and Ken Houck's laboratory at UCLA, really beginning to think about new ways that one can consider biological catalysts as a way to achieve site selectivity in late-stage CH functionalizations. Okay, next slide. Okay, I thought I would begin just with a few comments about some of the considerations to think about in site-selective CH functionalization. And it really informs the way that we think about the problems and where we viewed some of the gaps in the literature that we had hoped to address by this type of approach. So first one can think about substrate biases in a molecule, certainly things like hybridization in carbon, the bond strength and acidity, steric accessibility, stereoelectronic and inductive effects all become very important, really, regardless of the method that you're thinking about for site-selective transformations. That can highlight this earlier review from Newhouse and Barron that gave a really nice overview of some of these substrate-based influences that are important considerations to think about in this field. Okay, so certainly one of the big challenges in the field and one of the big interests throughout the CCHF is thinking about ways to use catalyst control to either supplement or override these kind of substrate biases. And I think first and foremost, there are really fundamental differences in the mechanism of CH cleavage processes depending on the metal substrate combination. These differences can be governed by the metal electronic structure, and they also can be very, very strongly tuned by the development of substrate ligand interactions that really become a key issue in the effectiveness of this type of approach. Dan, you can advance. I just want to highlight when I think about this, you know, I think it's useful to consider the vast array of different types of interactions that can be involved just in the individual CH cleavage step, not to mention the other aspects of the mechanism. So starting really from an agostic interaction, one can think about direct three-centered oxidative additions taking place. An acetate can participate in a CMD type of process, or an olefin can participate in the LLHT type process. And this latter area has been a big interest of my lab in the center work involving nickel catalysis, but that's a story for another day. Also, certainly there are many classes of reactions where things like metal oxos, nitrines, and carbines, where there's not an intimate association of the metal with the reactive CH bond, but instead some type of X group directly gets transferred to that CH bond. This can either go through a concerted insertion or through a stepwise addition followed by a rebound. And this latter pathway is thought to be the consensus mechanism of the cytochrome P450 enzymes that will be the subject of the lecture today. Okay, next slide. So certainly when these catalyst control strategies, they may not be effective for many desired outcomes, and thinking about direct in-group control becomes a very important way to consider this problem. So I would say typically these involve catalyst delivery strategies to the proximal CH bonds. Grumman gave some beautiful examples of actually the temporary connection without a permanent covalent attachment of the direct in-groups as a very, very powerful approach to this type of problem. One can also think about more extended templates, such as the U-meta functionalization work, that enable access to more remote regions of the substrate. So as shown in the center graphic, this really now, because of the design of that template, has an extended framework that enables the metal catalyst to approach more remote CH bonds within the molecule. And finally, in this latter graphic on the far right, I can just highlight that very extended templates have been described. Some of the beautiful early work from Breslow in the 1970s looked at these types of extended templates that could do directed reactions as extensive as going from the steroid A ring, reaching all the way across to the CD ring juncture methine CH by an atom transfer reaction under photochemical conditions. And so these are very nice strategies, but they have not been as well developed as a synthetic tool. Okay, next slide. Okay, so certainly thinking about biocatalysis now, this serves as an important complement to small molecule catalysis and can really achieve very different types of selective CH functionalization reactions. So I would say generally the enzymes can achieve the molecular recognition through an ensemble of weak interactions. but it's really the substrate generality and the reversal of site selectivity that I think poses the greatest challenge for this field in broader utility to synthetic chemistry. I will say, though, that tremendous success has been seen in the site-selective functionalization through directed evolution methods, and I'll just highlight it here, a beautiful recent example from center work from Francis Arnold and Brian Stoltz, looking at a late-stage oxidation reaction in the synthesis of nigeladine A. And so in this particular case, a variety of chemical oxidants were unselected among the four regions marked in the substrate, but in fact, by screening a library of BM3 mutants, they were able to identify a particular biocatalyst that was very effective at achieving the desired oxidation to complete the total synthesis of this natural product. Okay, next slide. Okay, so really where we came into this idea was that we wanted to think about using some of the capabilities of biosynthetic enzymes, but really marry this with the knowledge of directing groups that had largely come from the small molecule catalysis community. And so really the insights that led to the development of this collaboration came from the Sherman Laboratory, looking at understanding the biosynthetic pathway for the production of methamycin and neomethymycin, and also the two-carbon homolog picromycin. So in this particular example, YC17 possesses the two reactive CH bonds that are highlighted among many different types of CH bonds in this molecule. And the cytochrome P450 enzyme PIC-C is capable of oxidizing these two positions in about a 1-to-1 ratio to generate the natural products methamycin and neomethymycin. And so as highlighted in the crystal structure graphic shown at the right, this is kind of an unusual characteristic of this biosynthetic P450, in that rather than involving this composite of weak interactions, it really is a very dominant salt bridging interaction between the E94 glutamate residue and the basic amine of the desosamine sugar shown in blue. And so that salt bridging interaction forms a hydrogen bonded network involving the E85 residue, and that serves as a primary anchoring influence to enable substrate binding. And it's really very weak hydrophobic interactions elsewhere in the molecule that allow the molecule to glide towards the heme iron center, enabling oxidation of the two protons shown on the right in the substrate YC17. And so we thought this strongly directed influence in the enzyme active site was a really intriguing feature that might suggest that we could have considerable generality with the classes of substrates, since they're not really very important and very strong interactions with the oxidizable portion of the molecule, really just with the basic amine that's part of the desosamine residue. So we really wanted to ask the general question, can a single biocatalyst be used on a broader substrate range, also with opportunities to control and reverse site selectivity? So rather than looking at extensive variation of the catalyst structure, we're really going to try to achieve the selectivity and the site selectivity control by using the directing group approach. Okay, next slide. Okay, so really the first step in this project was described in this 2009 paper, and we recognized that the amino sugar desosamine was the key anchoring influence, and so we simply harvested that sugar from erythromycin. We could convert that into a protected form of the glycosyl fluoride and use that glycosyl fluoride reagent on the top right as a reagent for transferring desosamine to a variety of simple substrates. And we're very pleased to see, even by stripping down all of the functionality in the A glycone, we were able to see good binding and good conversion through a single point mutation, the D50N mutant of the PIC-C enzyme system. The bad news was that this was a very unselective reaction, leading to 7 monohydroxylated products, and so it was clear that, that some improvements on the regio control were needed in this general area. I'd just like to highlight that in earlier developments from the Sherman work described in this 2007 paper, they had developed a very nice system where this D50N mutant of PIC-C could be incorporated as a fusion protein with the RH-FRED system, which is a reductase domain facilitating electron transfer to the P450, and this system is then overexpressed in E. coli. And so we began with that as a starting point for the catalyst to really explore this type of approach. Next slide. Okay, so obviously harvesting and reinstalling these amino sugars was a little bit tedious, and so we wanted to ask the question about to what extent can that sugar motif be altered. And so we were pleased to see after a few iterations that this could come down to a very simple structure involving benzoic acid derivatives that possess a dimethylamino methyl substituent on the aromatic ring. And so in using the endogenous aglycone YC17, we could, from YC17, we could directly convert this using the PIXI catalyst to the same mixture of hydroxylated compounds. But perhaps most interestingly, we found that the regioselectivity could actually be altered by the precise structure of the benzoic acid derivative utilized in this transformation. So simply by moving that dimethylamino methyl anchor from the ortho to the meta position was a sufficient structural change to change the site selectivity of this overall transformation. So unfortunately, this was not at all a general observation, and in general, using these types of anchored systems with this particular P450, we could oftentimes see a single oxidized product, but we generally did not at all have good control in changing the site of oxidation, and so that was certainly a major challenge for us going forward. Okay, next slide. Okay, so what I've shown you then is that the endogenous substrate is effectively oxidized by PIXI. We can look at two independent changes. By looking at the endogenous substrate, we can go to synthetic anchors as shown on the top right, or by taking the native sugar desosamine and looking at an unnatural substrate as shown on the bottom left, that also leads to good conversions, albeit poor site selectivity. So we then asked the question, is it possible to really make both of these changes and do them in a more substantial way? So is it possible to take something like menthol that clearly is extremely different from the endogenous substrate, and then link that onto the simple benzoic acid linkers and see if these could undergo site-selective oxidation. So this is a major deviation from the synthetic substrate, has a synthetic anchor, and even though menthol is a simple molecule, it does present multiple oxidizable sites of methynes, methylenes, and methyl groups to the biocatalyst. Okay, next slide. Okay, so in the course of the study, this was about the point where we began to collaborate with the Hauck Laboratory, and they provided some extremely important insights to us in going forward with this project. And so in early iterations, thinking about the wild-type enzyme, they had looked at menthol linked to a very simple glycine-derived system possessing the dimethylamino group. And unfortunately, as you can see from the graphic at left, that the dimethylamino group of this particular substrate does not become well positioned with the E85 and E94 residues that were characterized as the salt bridging components of the biosynthetic pathway. Alternatively, this E246 residue positioned the substrate in an unproductive binding conformation and led to very poor catalysis for the system. And from these molecular dynamics simulations, this then suggested from the Hauck lab that basically looking at mutative forms of this enzyme could result in a number of important improvements in the overall catalytic activity. So, for example, mutating out that E246 and the D176 residues that led to the unproductive conformation, and then doing a second change by extending the anchor to now the benzene spaced with the para-substitution pattern as shown on the bottom, this now can restore the desired salt bridge and hydrogen bonding network on the upper left quadrant of the graphic shown on the right. And so these two changes now suggested that this triply mutated enzyme would be a much improved biocatalyst. This was borne out experimentally, and so this predicted observation really was a very important step forward in being able to address this type of approach. Okay, next slide. And just to show a few results with this particular triply mutated catalyst, you can see that the reaction works in a site-selective manner on menthol to get the product shown on the top left. It works in a site-selective manner with an unsaturated version, and you can do simple substrate variations looking at cyclohexenes, a couple of different benzene fused materials, and also a 2,2,1 bicyclic terpene, all of which gave a single major product of oxidation with good total turnover numbers in these experiments. So what I think you can take away from this slide is that these modifications have now enabled a wide substrate scope But really the limit in this chemistry, as I alluded to earlier, is that the site selectivity reversals were still completely lacking with these smaller terpene molecules. And it really raised the question about what can we do to better improve the capabilities for doing a directed reaction where we can change the site selectivity using a single biocatalyst in this type of operation. Okay, next slide. Okay, so the idea that we had was to really make some more substantial improvements in the nature of the directing group. And the way that we've approached that problem is we realized we needed something that was synthetically simple, that was highly modular, and that ideally would introduce the basic amine at the last step of a high-yielding reaction to really simplify the purification steps required in making these constructs. So as you can see, there are a wide variety of the azido acids that would be used for an initial esterification with the hydroxyl of the substrate. These can be directly derived from widely available amino acid derivatives. So with this azido ester in place, we can then look at click reactions with propargylic amines possessing different spacers between the alkyne and the basic amine. And as you know, this can be done based on the very nice work from Foken that either using copper catalysis or ruthenium catalysis, either regioisomer of the triazoles can be accessed. And so this is not just simply a matter of clicking together the oxidizable substrate and the amine, but instead the precise outcome of that click reaction really defines the geometry of that blue and red portion of the molecules, and it's really the shape and the size, the length and the conformation of that linker material that ultimately will be very important for identifying the relationship of the basic amine to the oxidizable substrate. Okay, Dan? And so the central hypothesis then is that by looking at a broad range of these anchoring components, changing the nature of the S1 and S2 spacers, and also the regioselectivity of the triazole, that we can establish the salt-bridge interaction with the basic amine, and according to the spacer groups that we've chosen, this will change the overall conformation and size of the molecule in a way that can present different hydrogens toward the heme iron center. And this was the basic hypothesis that we set out to explore in our most recent work. Okay, next slide. Okay, so with this in mind, we wanted to throw a really hard problem at these enzymes to see what we could do in terms of looking at the substrate scope and the site selectivity reversals. And so we thought macrocycles were a really interesting place to start. So as you can see with the two macrocycles that I've shown at the bottom, possessing 11 and 12 membered rings, they have 6 sp3 methylenes. There are multiple low-energy conformers accessible, and these two aspects really combine to give, I think, a considerable challenge for any of the existing CH functionalization methods out there. We were also attracted to this type of approach because it merged really well with some independent work that we were doing in our lab, looking at carrying out Regiodivergent reductive macrocyclizations of linear inals. And so the nice feature of this combined strategy is that you can start with a very simply, a very easily accessed inal derivative that's acyclic, and then to directly convert that into the 11 and 12 member ring products in a single step from that common precursor. So then that gave us a very nice handle for thinking about these two types of challenging substrates for the CH oxidation methodology. Okay, next slide. Okay, so the basic idea then is that starting from that simple inal, we can branch these into the two different macrocyclic frameworks, and then test the strategy of looking at the site-selective P450 oxidation reaction to really decorate each of these macrocycles with hydroxyls at different positions around the macrocycle. And so if you think about the traditional approaches to making macrolides, you're really looking at selective reactions involving carbonyl addition reactions by things such as aldol additions and alloboration methods. But this approach is very different, and that really from a single precursor, we can access two different macrocyclic frameworks that have a largely hydrocarbon-like framework. And then we're going to convert each of these to a subset of oxygenated products through the site-selective P450-mediated oxidation reaction. Okay, next slide. Okay, to show you a few results from this chemistry, we can start with the exomethylene compound that we made by an exocyclization. And actually looking at a simple panel of 12 of these clickable anchors, we could see that there were 3 different major products that could be accessed depending on the click anchor that we developed. So looking at a case with a benzene spacer that's paralinked, with this particular triazole, we could directly access the allylic oxidation product as the major compound. By looking at a much shorter framework derived from alanine in the middle, that particular anchor gave product 2, which involved oxidation alpha to the ester. And then thirdly, by looking at, again, a benzene-spaced compound, but now moving that triazole to the ortho position, that further changed the regioselectivity to favor the oxidation beta to the ester. So really, hydrogens in very, very different chemical environments could all be accessed from a single substrate and a single biocatalyst, simply changing the nature of the directing group as the key component that alters the regioselectivity in this type of reaction. So you can probably imagine that making predictions about the site selectivity is not at all intuitively obvious based on these types of approaches. So we're really interested in a more predictive and more rigorous mechanistic analysis of how this chemistry was working. Okay, next slide. So to really accomplish that, the HALC lab has done a beautiful job looking at a combined DFT-MD approach to understanding the site selectivity. So I'm not going to do a lot of comparative work with different substrates. I'd like to just sort of walk you through this computational workflow in a single substrate to show you the general approach that has been used. So in thinking about one of these substrates that has the parabenzene spacer anchor as shown on the top left, basically the process that one must go through here is to first identify the low energy substrate conformers. In this particular case, there were primarily 2 different conformers that could be accessed, with multiple conformers being significantly higher in energy that could more or less be ruled out. So once those conformations are identified, then the individual inherent DFT barriers for the CH abstraction were measured, sorry. And so the way that this was carried out was that the macrocycle was considered in a truncated form possessing a simple methyl ether in place of the linker, and the P450 biocatalyst was considered again as a truncated form without considering at this stage the active site features. So once those individual DFT barriers for each of the different CH bonds was computed, then it was time to really look at the fully functional system. So by putting the linkers in place, considering the entire enzyme active site, one can then look at the molecular dynamics approaches over a 500 nanosecond time frame to basically consider which conformations in the active site are accessible and which CH bonds are in proximity to the heme to enable oxidation. And so specifically, the HALC lab looked at the distance and the angle of the substrate CH bond to the biocatalyst iron oxide unit, as you can see on this far right graphic. And so basically what's done here, you can see the purple and yellow graphics from this molecular dynamics plot, and this corresponds to the 2 protons that are highlighted in the top left structure that indicate plotted the distance on the horizontal axis and the angle on the vertical axis. And shown in red at the top left is the idealized transition state geometry. And you can see over that 500 nanosecond time frame which of these methylene protons most closely approximate the idealized transition state geometry for the CH abstraction. So you can take that information, then compare that with what I described in point B, which is looking at the inherent DFT barriers for the CH abstraction. And using this protocol, they can very accurately predict which CH bond in these types of substrates will undergo the site-selective oxidation reaction. So it's a very important predictive tool that can be used in combination with the high throughput nature of the click anchor assembly. Okay, next slide. And so just to highlight a few of the other subtle differences that become very important here, the examples that I showed in the previous slide using this particular paralinked anchor produced the allylic oxidation product as the major product of reaction. Simply looking at the enantiomer of this structure, with the same biocatalyst actually leads to a different major product, now leading to the oxygenation alpha to the ester. So it clearly is a matched influence between the substrate chirality and the overall chirality of the enzyme active site that becomes a very important influence in dictating the site selectivity of this type of transformation. And so furthermore, by taking that top substrate, converting this to the epoxide through chemical methods, this does two things. It deactivates the allylic, what were the allylic protons for activation, and also adjusts the overall conformation of the macrocycle. And this now also leads, rather than the allylic oxidation, of now producing the oxygenation alpha to the ester framework with, in this case, very high selectivity. Okay, next slide. And just like to highlight quickly that, as I mentioned, we can take these same inal precursors, change the ring size by altering the nickel catalyst that is used in their assembly, and then now taking the 12-membered ring structure from this same precursor, we can see oxidation at either the allylic position or beta to the ester functionality using a pair of the click anchors that were identified in the previous study. Okay, next slide. So really, just to summarize, I'd like to highlight that this has been a progression of influences, starting with the endogenous sugar, desosamine, that can be an important influence in developing these anchoring effects. This could then be extended to thinking about simple benzoic acid systems bearing a basic amine that can lead to a very nice expansion of the substrate scope. It was really this approach of looking at the click anchor methodology that really brought the tunable site selectivity into the mix and allowed us to make a range of different oxidized macrocycles with differing ring sizes, all dating back to a single precursor of this type of inal that's shown in the middle of the graphic. So in this case, we're now able to use a single biocatalyst, have pretty good substrate generality. We now have tunable site selectivity for the reaction, and I think a very nice detailed mechanistic understanding come from the computational work of this type of approach. Okay, next slide. Okay, so I'd really like to thank overall the CCHF. They've just been a fantastic group to work with and to collaborate with and to learn from, and I've really enjoyed the time that I've been able to spend with them, and thanks again to Hugh and Dan for putting this symposium together. On the bottom graphic, I'd like to thank my group. They mostly do work very different than what I talked to you about today, but they've been a terrific group to work with, and it's really been fun to integrate the projects in biocatalysis with the more with the organometallic chemistry that most of my group is currently looking at. Okay, next slide. So I'd really like to thank the team of students over the past 9 or 10 years now that have looked at various aspects of this collaboration. They're all listed from the publications that were cited along the talk. And I also wanted to highlight the 4 individuals in bold: Michael, Matt, Jessica, and Song, who have really been key players in the most recent developments of the unpublished work that I described in the latter points of the talk. So again, I'd like to thank Hugh and Dan for the opportunity to present today. I'd like to thank the funding agencies that have supported different aspects of this work that I've talked about, CCHF and NIGMS and the Rackham Graduate School at Michigan. And if there's time, I'd be happy to address any questions. Thanks very much.
Thanks very much.